Joey is the program director for Forest Health Watch, where they empower communities to keep forests healthy. IATC has had the pleasure of working with Joey on many occasions. This is our second webinar together, and Joey has generously joined us on a number of hikes, teaching us about Western red cedar dieback and how we can help with iNatural documentation and soil sample collections. Since Joey taught me how to identify unhealthy cedars, I am continuously looking up. Sadly, as I look up, I'm finding more and more dead cedar tops, and my commitment to support this work grows even stronger. And Joey, I would also like to congratulate you on your new, your 5-month-old baby girl. Thank you and congratulations, and it's all yours.
Excellent. Thank you so much, Anne, and thanks, Nicole, for having me and including me in this. And again, just like to echo what Anna said, it's been a real joy to engage with this community, and I would like to do so more often as we go forward. So please don't hesitate to reach out. Happy to get involved and join you on a hike any day. And you'll also see that some members of IATC have actually come to Puyallup and visited us and dropped off some soil samples in the past. So we certainly welcome visitors and would love to have any of you come and tour or organize with me to come and see some of the lab behind the scenes in the lab. So Nicole, if you could just confirm that you can see my screen okay? It looks okay?
Yep, looks great.
Okay, wonderful. So yeah, thanks so much for spending your Tuesday night with us. It's again really a treat to be here and to share something that we're really passionate about. We love Western red cedar, and we love community engagement, as Anne said. So this project has been a really fun opportunity to explore both. But as Anne said, it is kind of disappointing or discouraging to see so many unhealthy trees. But I'm hoping that tonight you'll learn that despite the challenge we have ahead of us, there's a lot of opportunity to work together and to learn from each other. So on that note, we love to engage communities, not just as a means to collect more information or more data or more soil samples, but to learn from you. So I'd love to hear your perspectives and hypotheses or your own stories about red cedar as we go forward. So tonight, we'll talk about red cedar dieback, the issue, and then some opportunities to collaborate on some soil sampling to investigate the within site variability, if you will. And I'll go into this more, but generally, very often we'll see a healthy tree next to an unhealthy tree. And we have some questions about what's going on there. If it's a climate signal, why aren't all the trees affected the same? And so there's some opportunities to explore that a little bit further. I am a researcher at WSU, and I'm based in Puyallup and Tacoma, but we have support from WSDA, and we just finished a grant from DNR to work on a maple tree disease. So together, there's a few different agencies that have provided support for me to be here with you and to share with you this information. I'm going to briefly introduce our program in Puyallup, but then we'll talk about red cedar and the red cedar issue. And we'll talk about how some community scientists have contributed to our understanding of what's going on, and as well as some next steps to really investigate what's going on within these sites or within some of our landscapes. I do want to note that I'm really privileged to live in the Puget Sound, as many of us are. It's just incredible space that's been occupied since time immemorial by many Coast Salish nations and peoples, and we are really privileged to do everything, a lot of learning, exploring, discovering on kind of in this region, the traditional homelands of the Puyallup tribe Indians. But it's really nice to be able to share some of that knowledge with you all up in the kind of the areas of Snoqualmie and Skokomish, and Muckleshoot. So if you haven't explored this tool, nativeland.ca, it's really, really neat for understanding whose homelands and territories you may be present or visiting. So as I mentioned, I'm a researcher at WSU, where we have a research and extension center just east of Tacoma and Puyallup. And it's really old research center, but we're really privileged to have some space to do some research on the ground. So we have some trials with Christmas trees and madrones and ash trees. We're looking at genetic diversity, but we also have greenhouses and labs and other spaces to do some work. There I'm mentored by a number of great people. I'm not just a single individual or individual researcher. There's a team of us. But I do want to just kind of step back and talk about where my background. So I grew up in Kansas, and my family moved to Washington about when we were studying, about when I was going to start undergraduate. So at that point, I decided I had to study trees because we were moving to Washington. So I got a bachelor's in forestry at Washington State, and then went to Oregon and studied a tree disease called sudden oak death that I'll talk a tiny bit more. And after that, I went to South Africa and did a PhD. And there again, I studied plant disease. And I was fortunate to return to the US in 2020 in February, just before things started to lock down. And since then, I've been a postdoc at WSU in Puyallup. Our program in Puyallup also studies a lot of different tree health issues. I mentioned that there's a number of people in the program that mentor me. One is an expert on true fir diseases and Christmas trees. Another works very closely with Pacific madrone trees, so arbutus. And together we all study tree health issues. So this is sooty bark disease. We work with nurseries to bear with me while I turn off this timer. But we work with nurseries to reduce spreading invasive species in nursery trade and trade of live plants. We also study a number of invasive diseases like sudden oak death, which you can see here in the top right. Sorry, now you can see. So this is a tree health issue caused by a Phytophthora. It's predominantly in Oregon and California, but occasionally we'll find incidents of this organism causing this disease in Washington, particularly when we plant new plants from ornamental nurseries or nurseries buying or trading materials with California, Oregon, or British Columbia. More recently, we've really started to focus on tree health issues in the urban environment. And we were given a very generous gift by the Albert Victor Ravenholt Fund to start this lab and focus more on tree health and community health in the intersection there. And so we've been working around Tacoma, looking at a maple disease, and as well as trying to look at the dieback of red cedar across our urban landscapes as well. If you yourself, or if you know of any students, we are always looking for more opportunities to engage with students, and we welcome more opportunities for more students to do internships or projects with us in Puyallup. So please keep us in mind if you think any of this would be interesting to any students or youth that you engage with. So that's a quick introduction to our program. I'm happy to elaborate more on any of that if you like. You're welcome to post any questions in the chat, and I can try and address those as we go. You're also welcome to raise your hand or just speak up at any time. I won't be bothered, but maybe for the sake of the recording, it's best if you can just post it in the chat for now, and then we'll have some time to discuss relatively soon. I do want to spend a few minutes highlighting why red cedar is so special. It's probably arguably one of the most important socioeconomics trees in the Northwest. So when we think about the cultural importance because of the legacy that it's had in Northwest people's lives and the livelihoods of Northwest peoples, but also in the kind of the establishment of the Northwest as timber capital of the world during the industrial ages. So red cedar often grows in these kind of, well, I guess it's not very often that you find a completely dominated stand of Western red cedar, but it does exist. This photo is from Anacortes. And so occasionally you can find pure stands of Western red cedar, but actually the highest density of Western red cedar is actually in kind of the interior distribution. So in northern Idaho, in this very, very western Montana area where is where you find the most kind of dense stands of Western red cedar. But you can see here that Western red cedar does have a very large footprint on the landscape. So we know that red cedar is really, really important because of the space that it occupies in the Northwest. And there it's either existing as a codominant with other species, or some cases it's the dominant tree in that stand. Here you can see, this is from a couple of years ago, but Western red cedar is very commonly mapped on iNaturalist. And it's neat to see that the distribution ranges all the way from Northern California, and it goes further up all the way into Southeast Alaska. Western red cedar is really important culturally. We know red cedar is considered, well, it's provided many different gifts, so it's considered a really generous species. So red cedar, in this case, you can see from these historical society images, red cedar is used commonly for textiles, weaving baskets, weaving clothes, weaving red cedar hats. It's also used for shelter, creating these mats to create shelters, and of course it's used for canoes by many of the Coast Salish peoples that were active in fishing and kind of migrating or transporting via waterways. So red cedar is also really commonly used. It splits really easily, so it was established. It was fundamental for these long houses or these plank houses, where these very long straight planks could be created from Western red cedar to create these long houses. It's also used ceremonially and is used for carving. But generally Western red cedar, again, is considered really generous species because of all the gifts that it's provided to indigenous cultures since time immemorial. You may have also heard red cedar referred to as the tree of life. Again, red cedar has many different uses, but I guess the saying is that from the time that you're born, you benefit from Western red cedar to the time that you die. I've kind of learned a lot from studying Western red cedar on a personal level in terms of whether it's appropriate for me to share indigenous knowledge. But I was given this cedar, initially I would tell people what this cedar bough is given to me for, but now I've questioned whether it's appropriate for me to share that knowledge or not, because I feel like one of the things I've really learned from studying red cedar is that this traditional knowledge is earned. And so for me to share, that this was given to me as incense for ceremony or medicinal uses, I question now whether that's appropriate, whereas before I would share that willingly. And so that's just an example of one thing that I've kind of learned from studying red cedar, if you will. Another neat thing about red cedar is its role in our industrial legacy in our timber towns like Issaquah. Certainly was probably a mill town before, became a really awesome tourist destination, now a recreation center. But This picture in the bottom left is a picture of the Ballard mill with a lot of red cedar shakes, or in this case, planks. But these in the top right, these are what they're called shakes, or they're essentially shingles in some cases. So red cedar was very commonly used for shingles on housing, but we moved away from that because of fire hazards, of course. But red cedar was a huge part of kind of this booming Northwest culture, and a lot of the establishment of a lot of our towns, which are basically timber towns or mill towns. Still today, it's considered an enduring wood of the ages because it's highly durable and it's pretty rot resistant or decay resistant. So there's still specialty mills that create products like fence posts and other, other boards and materials that you use for your decking or your fences or other areas where you need that durability. So, for example, a raised bed or something like that, garden bed. Red cedar is also really critical when we think about the habitat in the Northwest. Previously we were talking about pure stands of Western red cedar, where it's critical for habitat of species like owls. And it's also often the species that we find growing along our rivers and streams, where it's providing exceptional shade to those stream banks for salmon and keeping those streams cool and healthy so our salmon can spawn. I love to share these photos as well, just because red cedar commonly is in spaces that we gather, and we often gather around red cedar or huddle under red cedar in the rain, and it's just neat to think about how much of a footprint this tree has in our lives that maybe we do or do not notice. But I did share a little bit about how I've learned from red cedar. Generally, I find myself in kind of these ethical dilemmas of whether, in the face of climate change, do we hold on to these trees that are struggling, or do we move on and try and adapt? But frequently, when I reflect on the health of red cedar and the role of red cedar in indigenous cultures for many, many generations, it's hard to even consider moving away from it and trying to plant other species. And so on a personal level, I've had to reflect a lot just from recognizing how important this is and learning about the amazing kind of the central place that this tree has been in a lot of communities and a lot of lives. So red cedar is really, really important, and it's something that we need to understand very carefully about what's going on and what issues are affecting red cedar. One, it has a really big distribution, so it's going to have a large impact if there's something affecting red cedar. Two, it's really culturally important. So will future generations have access to the same gifts that many generations beforehand have had, right? And unfortunately, we're starting to see this die back, and I hear different estimates on when this started a lot. Some folks tell me that they've been watching red cedar die back for 10 years or so, but a lot of the reports and kind of headlines and things have really started since 2015, when Northwest experienced a pretty substantial drought. But yeah, every year we still see new and like, if you probably around Issaquah right now, you'll find trees that are just now turning brown and having these health issues. So it seems like the issue is continuing. It's not really like a one-time drought event that we've seen. There are a number of trees that look like this throughout the Northwest, really ranging from British Columbia down to Oregon. Particularly in kind of William Valley North and Skagit Valley and these lowlands, you'll see these trees with dying tops or thinning canopies where it's really easy to see through. And I think a couple of these pictures have probably been taken on hikes that we've done together. But there is another group that has recently shared this kind of preprint, if you will, of a paper they're submitting or they've submitted. This is a really incredible group. It's led by Robbie Andrus. Robbie's a postdoc like myself at WSU as well. And he spent a summer coring trees. So they would trick these tree cores, and with those he could look at the growth rings of each year. And what was unique about this study is they cored trees that are healthy and trees that were unhealthy, and they looked for differences. And so initially they found a kind of a climate relation with tree growth for healthy and unhealthy trees. But the difference between the healthy and unhealthy was the consecutive droughts. So what they noted in this study is that if a tree experienced drought like 3 years in a row, that's when you see the dieback, instead of saying it's not just one event. But they suggest that and often the trees could rebound if they had a drought event, but then the next year was a wet year. And so in many cases that did explain why trees were still healthy. But in areas where you had this kind of repeated compounding drought is where you start to see this dieback. And so they would core trees. Again, they would core these dead trees, unhealthy trees, and healthy trees, all from the same sites. And they would look for kind of these differences in the climate signal. And what we really learned from this is like red cedar is definitely struggling with the longer and hotter summer droughts that we're experiencing. And the more droughts we have, or the more continual, repeated droughts that we have, the more our trees are going to struggle. And it's really a tragedy when you think about how important red cedar is to indigenous peoples. And we've been encouraged to think about the dieback of red cedar as similar to having a relative that's sick or having a language disappear. And so it's really not as simple as just moving away from red cedar and trying to adapt to a hotter or more frequent drought. We need to explore opportunities and maybe genetic diversity and moving some seeds around. We need to do some experimenting before we completely give up on this tree, because it's so important. And so often we'll see this, I'm doubtful of their success, and I've chosen other trees to plant on my property. In many cases, this is smart depending on what your objectives are, but there may be places where red cedar could be protected or it could be monitored. And then there may be other places where we could restore red cedar. And there's some questions that we'll go into now about kind of the within site variation in a minute. That also draw some questions, and it's not just as simple as if they're doing bad on my property, then they're not going to do good in the future, or we need to move away completely. So we've been really reliant on community engagement and folks like yourselves to share observations. And with this, we've been doing some neat analyses, and we're trying to be as inclusive and open about and transparent about our analyses as we can. So we welcome anyone else to get involved in the analysis of this data. And I'm happy to share more about that. We've been working with some great folks lately, and I'm excited to share some of that. We also like community engagement because from the standpoint of invasive species or insects or diseases, it's great to have more people involved, and we really think about keeping our trees healthy is a shared responsibility, and we need more people to be involved than just say the federal government or our urban foresters or our researchers. It needs to be kind of a community effort. And so that's really what's led us to initiate this program called the Forest Health Watch. And it's been a really incredible program from the lens of the community engagement. And we've really, really enjoyed working with so many people and learning with so many people. With this program, the first step, when we first started this project, we asked all our stakeholders that were engaged and our partners, what is the priority issue? And it was very clear that red cedar was the biggest concern to folks because their trees were actively dying throughout the Northwest. And so at that point, we tried to figure out how are we going to work with communities and trying to advance and accelerate knowledge about this issue. And so we started to look at iNaturalist, and we really like iNaturalist because of the number of people that are using it and because of the stability of the platform on the web and on your smartphones and mobile devices. So generally how it works is you can share an observation of any organism, and the community will help you identify what that organism based on the pictures you share. And in our case, rather than discussing the findings, we've created a project that people can share the observations to. And when they do that, it asks a few other questions. So I'll share a little bit about what that looks like. In case you are new to the mobile world and the power of these cool platforms, iNaturalist has also created an app called Seek, which essentially, by all the people identifying organisms and sharing observations, it essentially made a really good machine learning or AI, if you will, or an algorithm for identifying organisms from your camera. So this cool app called Seek, you hold your phone up to a plant or a butterfly or an insect, and it will do its best to tell you what it is. It's pretty powerful. It's really good at identifying. It's pretty amazing how good it is at identifying insects, but it's good at identifying plants, especially if they have flowers or fruits. They're not so great when there's, when there's nothing charismatic about the plant. But I do want to just note kind of how the iNaturalist project works on from the Red Cedar Dieback Map project. So when we first decided that red cedar was the focus because so many people were concerned about it, then we identified iNaturalist as a tool, and then we worked with these great folks from Department of Forestry and Forest Health and Protection and the Washington Department of Natural Resources to develop the project into something that would be useful for them, as well as something that kind of expands on the work that they were already doing. So we work with them to make this project in a way that asks questions that would be valuable to them. This is slightly outdated, but this is kind of where we are today. We've got just over 2,300 observations have been shared. So these are all individual tree points. Nearly 300 people have contributed, but I think like close to 600 people have joined the project. So it's really been neat to have this kind of community come together and add so many observations. And it's really been neat to see some of the super contributors that have gotten involved and shared more than one observation or shared as many as 250. This gentleman, Jim, which is incredible to have this dedication and passion from community members. There is a guide for contributing to this on foresthealth.org/map. You can download a field guide, and I'll try and print some and bring some to the hike on Friday. But this guide just takes you step by step on how to use iNaturalist. You also can take pictures from your camera, and then when you're back home on the computer, you can add them. So you don't have to use your phone if you prefer not to. Someone was just messaging me yesterday about how much data it uses because they were concerned they don't have that much phone reception or data. So some cases it might be easier to just take pictures and then add them back on your computer. We also have this neat dashboard on foresthealth.org/analyses, where you can interact with the data and see where dead trees have been shared or trees with healthy trees or unhealthy trees. What's neat is that we are asking folks to map both healthy and unhealthy. And that's been really, really helpful for the type of analysis we've done, which is trying to find the parameters that classify trees as either healthy or unhealthy. So generally how this works is every single time someone has a observation, there's usually a GPS point, and most often it's pretty accurate. If you don't use your phone, you just kind of drop a pin where you saw the tree when you're adding it on your computer. But generally, once we have a GPS point, there's so much you can do with that information. So we use that GPS point to collect climate data. So they've downscaled kind of these big global models so that you can actually get a different climate variable for this 4-kilometer grid cell compared to the one 4 kilometers away. So it's pretty powerful. So each of these points can have a bunch of different climate data. And then we've also been exploring some of the soils data. And so we're interested to know, is there certain soil properties that are associated with healthy trees versus unhealthy trees? And we'll talk about that a little bit. And then the other piece we can pull in is from a GPS point, you can get neat things related to the topography. So you can figure out like what direction is it facing, how steep is the slope, generally like how much sunlight that spot gets on a given afternoon. Those kind of things are pretty powerful just from these GPS points these days. So once we have all of that information, we try and predict where trees are healthy and unhealthy, or we try and identify which predictors or parameters are important for classifying that tree as healthy or unhealthy. And so that's been really neat. We've been exploring this approach with, it's an approach called random forests, which is again like a machine learning technique where you just throw all this data at a computer and you say, okay, tell me which data is the most important at breaking up healthy trees from unhealthy. And so commonly when we do that, it creates a classification tree like this, where you can see the variable on the top is the most important splitter, if you will. So it splits the healthy and unhealthy trees the best. And so water table depth is very often very important predictor or parameter for explaining or classifying if a tree is going to be healthy or unhealthy. So when we throw all that data at the algorithm or at this machine learning approach, it tells us which parameters are the most important. So generally it's telling us like soils is the most important, and then it tells us which climate variable is really important. And so we've been looking at all the different climate variables we can pull in, like moisture or precipitation or temperature, or the combinations of the both, like this index called the climate moisture index, which is like how much precipitation there is versus how much evaporation there is. So it's a combination of heat and temperature. But anyway, in the summary of this is when you throw all these data at it, it's actually pretty good at splitting up the healthy trees from the unhealthy trees. And that's been pretty neat to explore. And so we do want to get to a point where we can build some maps and actually predict or like give landowners the tools to make decisions about where to restore red cedar or where to protect red cedar. This is another example of a soils parameter. It's weird. It's part of the soils data set, but this parameter is called water table depth. And so unfortunately, like we're realizing that this soils data is not the best because out of the 2,300 observations, it only had data for 600 of those points. So we kind of learned that we can't use this as much as we thought we would, but we liked it because it was it separated the healthy trees from the unhealthy trees pretty clearly. And so this is suggesting that the lower the distance from the surface to the water table, the less healthy the trees, essentially, or not, the more unhealthy trees. And so it's actually not very intuitive. This is saying like shallower water tables had less, or most of the unhealthy trees were in the shallower water tables. So yeah, so it's not straightforward, but so imagine you're on the surface, you're standing on top of the soil, and then the depth to the water is what we're talking about. And so the shallower the depth to the water is where the unhealthy trees are. So it's kind of backwards, where you would think that deeper water table like would have, so in this case, the healthy trees are in areas where the water is deeper in the soil. And so I encourage you to think about why that might be. We've got some hypotheses, but generally it seems like the trees in the water, if you will, are less healthy, or there's more of those trees are less healthy. We're going to have to discuss this more because I'm not doing a great job explaining it. So I encourage you to keep your questions in mind, and let's discuss it more, so make sure we're all on the same page. But when we're out on a hike on Friday, we can think about this more. Like, why are the trees next to the water less healthy? Or why are the trees in soil that's typically really wet less healthy? I'm going to skip this. Yeah, so I see Sally's already got a great hypothesis. We can talk about that. That's definitely one angle. Sally's commenting like, okay, maybe if they're in more water, then they have more fungi. That could be part of it. The other aspect we can talk about is generally, if they're in really wet soils, they haven't had to put their roots very deep. So then they're more vulnerable to those water tables drying out because they don't have very deep roots. Is another thing we're thinking. But let's keep this in mind. I just want to say, like, with this data so far, this is kind of our approach. We'd like to be able to build some maps that stack all these different important parameters on top of each other and tell you where a tree is likely to be healthy or where a tree's likely to be unhealthy. And so we can make some decisions about planting red cedar going forward. That's the dream. We submitted a statement of intent to apply for a grant, and we're just kind of wondering if they're going to invite us to submit a full proposal to really dive into that and try and produce more of a tool. But we're slowly working on it, kind of on the side as we go, and that's part of the reason we totally welcome anyone that wants to get involved in these analyses to join. So one thing that we haven't really talked about is this kind of within site variability, and this is somewhere we'd like to go with the soil samples, and like to team up with you all to investigate. Very often you'll have a tree that's right next to each other, a tree that's healthy versus a tree that's unhealthy, and one way that we want to visualize this is talking about, what's going on, what's different between the tree that's unhealthy, that has dieback, versus the tree next to it that's healthy. And so again, we know that there's this kind of climate signal going on where these trees are, increasingly stressed because of our longer and hotter droughts, especially if it's repeating and compounding. So if every summer this one area has a longer summer or a hotter drought, those trees are going to be in trouble. But in addition to that, or if that was the case, you would expect all trees in that site to be struggling at the same level. But what actually ends up happening is we'll see one tree that starts to die, and then a few years later, maybe we'll see this other tree start to die. But it seems like each tree is affected by climate at kind of a different rate, if you would. So what are the factors that explain, why this tree in the middle is dead and these ones on the outside are still alive? Or, what's going on with this tree compared to this tree? What has this tree done differently to tolerate that dieback issue? And so we kind of think about this like within a site, there's a lot of heterogeneity, or there's a lot of variability between the tree health. And we want to kind of understand what factors are contributing to that variability. And so this is where we really are starting to think about kind of, well, there is this overall environment signal where the trees are becoming more stressed from longer and hotter droughts. And we know that we're talking about red cedar, and there may be some variability in the genetics of each individual tree. And that could be why some of them are healthy and some are not. And so maybe, another step would be to start collecting seeds and collect seeds from the trees that are doing well versus the ones that are doing, poorly, and start comparing those in different controlled experiments. But another factor that could be involved is the presence of root diseases or other pathogens. In addition to these, maybe you'd consider, environment. There's also other factors like the distance that tree is to disturbances from roads or root compaction or pollution. It could be related to proximity to the water, or it could be related to the amount of sunlight that individual tree gets, right? So all of these kind of contribute to that variation within a site. But what we're really interested in investigating with you all is kind of the role of plant pathogens and root diseases. And so this is a site on the south side of Sauk Mountain, where there's a number of dead red cedar trees. And when we looked closer, you could peel the bark off, but we found evidence that armillaria was playing a role. And so armillaria is a fungi that it's pretty common in our forest, but typically red cedar is thought to be pretty moderately affected. It's more common on other species like Doug fir and some of the other, I don't know about hemlocks, but I'll bring this awesome guide when we do our hike on Friday that kind of compares tree species susceptibility to this armillaria species. But generally, when you see these white mycelial fans, it's what we call those, like, that armillaria has colonized that stump or that tree. And what's unclear is whether this, killed the tree or whether it came in after the tree was dead, because it's kind of an opportunistic fungus. But generally, when I see, a handful of trees in a cluster that have all died and one of them has armillaria, then I'm pretty certain that this may be the culprit really affecting these trees. And we don't have a good understanding of like how many of our trees are affected with this, and how many of the trees that are dying that we see all the time are actually just dying from armillaria versus climate. And what is that combination? So again, armillaria makes these white mycelial fans. It's sometimes called the shoestring fungus because it creates these kind of rhizomorphs, these shoestrings. And on the right conditions, it will actually make mushrooms called the honey fungus, because apparently it's good tasting. Another group of root disease pathogens that we're interested in is called Phytophthora. There hasn't been any reason to think that Phytophthora is affecting Western red cedar, but it is kind of a group that's notorious for causing issues in natural forests. And in some cases, there's been invasive species that are introduced. So it's something that we want to definitely watch out for. So this, for example, is a Phytophthora species that affects Port Orford cedar by causing Port Orford cedar root disease. So this was a Phytophthora that was introduced to Central Coast Oregon near Port Orford, and they've done a really, really active role, played an active role trying to protect big old Port Orford cedars from getting infected. But it's generally moved by people and soil and things like that. Often if you see, sometimes other people call them Lawson cypress. They're planted ornamentally all over the world, but if you have a red cedar, sometimes we see sometimes we see cedars dying in our urban environments, but it's actually this Port Orford cedar, not our red cedar. And if it is Port Orford cedar, then it's likely dying because of this pathogen. However, this one only attacks Port Orford cedar, and it's not known to, I think it can kill yew trees, but it doesn't affect any other conifers in the Northwest. And I have this picture in the bottom left because Oregon State University has a really robust breeding program where they've bred resistant cedars to this disease. So that may be one option going forward if we found a root disease issue on our red cedars. Phytophthora is also well known for causing sudden oak death. So this is a landscape with a lot of oaks, tan oaks in this case, where all the gray are dead for a while, the red are just dead, and then all these lighter green are going to die because of this terrible invasive Phytophthora species that was accidentally introduced to California. So again, that's just two examples of Phytophthora. And so both of these are kind of setting the stage for what we should, maybe we should be looking a little harder to see what Phytophthora are in our forests, and then whether there's any possibility that they're playing a role in the dieback of our own trees like red cedar. So we've done a little bit of sampling. We've collected samples with IATC on two hikes, actually, two different hikes both on maybe one was on Sauk, but anywhere in the Alps.
Tiger and then Squawk. You got
Okay,
it.
thanks, Anne. Yeah, so we collected some on Tiger and we collected some on Squawk. This is some students from Green River College who did a study on the south side of Green River in their school forest. And what we'd like to do is collect more so that we can continue to get a better kind of sampling distribution and better understanding of the factors affecting our trees. But Phytophthora are really unique and they produce these spores that can swim. And so one way that we collect Phytophthora from soils is we place the soils underwater, or we soak them with water. We put them in these trays, we fill them with water, and then we float leaves on top of the water. And these leaves are like bait. So we're trying to catch the Phytophthora with these leaves. And so if Phytophthora is present in the soil, it'll swim up, the spores will swim up and affect these leaves. And so occasionally you'll see kind of these black spots emerge where they've been infected by Phytophthora or a closely related organism. After that, we cut small sections out of the infected leaf and we put them in the Petri plates, and that's where we observe what's growing out of the symptomatic leaf. And so in this way is where we kind of isolate all the different possible Phytophthora in the soils that we collect. And so at this point, we've kind of developed a method to do this, and we've collected samples from a few different places. And I'll come back to this. I'll come back to this guide in a minute, but I do want to just share some of the results. We've kind of done two different stages. So initially when we did the samples, I think this included the Tiger Mountain samples and the Green River samples. We had about 100 samples. We found 15 of those samples had a Phytophthora species. And so that's pretty, that's 15% of the samples, essentially. It's not very much, but it's hard to say if that's a little or a lot because we're only collecting a tiny bit of soil per tree. But what's been interesting is the diversity of things that we're finding in these soils. And so we found a number of different Phytophthora species in this first round. We've kind of repeated this. Sorry, I'm just going to go through. This is actually an underrepresentation of everything we found. We found 1, 2, 3, 4, 5, 6, 11 different Phytophthora species in the soils that we've sampled. These are only the species, the actual isolates that we've sequenced. So there's more that we grouped as Phytophthora, but we didn't sequence everything because it gets expensive to sequence everything and time consuming. So we're still in the process of kind of paring down all these results, but it does seem like there's a lot of different species out there, and in many cases we have only found some of these species like one time, so it's unclear whether they're actually important or playing a role. But there's a few like Cambivora that we find a lot, or Plurivora. And so with these ones that we're finding more often, we need to do some actual experiments in the lab where we're infecting trees to see if these species are actually harming the red cedar or not. They might just be present in the soil, perhaps causing issues on some of the other trees or some of the other plant species that are there, but it's unclear. And I just have this like small screenshot to highlight that some of these folks, Marilyn and Dave, brought some samples to us. We found Sue's syringe. Anne has a number of samples on here. Thank you, Anne and Marilyn. Where with Anne, we found a couple things like Plurivora and Cambivora and Cactorum. So there are some species out there that we need to do a lot more research on, but I will say that so far, we're not finding them very often, and it doesn't seem like we're finding them on unhealthy trees only. We're also finding stuff on the healthy trees. So really, we need more kind of deeper investigation into this to understand if they're playing a role or not in the health of our trees. But it's been a neat opportunity to go and kind of explore some things going on in our forest, and it's really a kind of wide open group of microbes that we don't have a good understanding about. So in that case, there's a lot to understand more about the role of these pathogens in relation to the dieback, especially in combination with the longer and hotter droughts. But the last thing I wanted to share about this is, there's still opportunity to explore possible solutions with the hosts. So not necessarily moving away from red cedar, but looking at different seed zones. And so we do have kind of a neat experiment going where we have 3 different sites. One's in Discovery Park in Seattle. One's kind of in this Black Lake region of Renton, and then one's down in Tacoma, where we planted red cedar from Oregon next to red cedar from Washington. And we're monitoring over time how they compare, how they do. And so we've just done 1 year of measurements on those. So we have, the trees are only like a meter tall or a couple feet tall. But we're interested in are the trees from Oregon going to do better than the trees from Washington, or what will happen? So that's the summary we have tonight for the issue with the red cedar and why we'd like to work with you to collect some more soil samples. I do want to just say that I'm really, really appreciative of this group, and I would like to think of some projects together that you all would like to do, and we could create some projects to work together on. It'd be neat to think about some hypotheses that we could test together and work together to collect some samples. Before I open up to questions, I did want to just share that guide for collecting soil samples. But before I jump over to that, I do want to encourage you, if you want to share your feedback or suggestions or research ideas or whatever you think we should be doing, there's space to do all of that on our foresthealth.org/share on our webpage there. And you can do so anonymously or not. But I did want to just share this. If you go to foresthealth.org/soil, you will find a soil sample collection guide. And so this just provides some steps, kind of some basic background on what we're doing. And if you're willing, it would be lovely if you wanted to collect some soil samples before Friday from your own property or from somewhere you go adventuring, or anywhere where there's unhealthy trees. But if you bring them on Friday, then I can bring them back to the lab and include them in our next analysis. But on Friday, I think the plan is to do a little hike and then also go and collect some soils together, so I can demonstrate this all while we're there, and we can collect some soil samples too. But it would be great if you are willing to collect some samples before Friday and we can pick them up. So this guide will kind of show you the steps, talks about the materials you need. Generally, we just collect soils into a Ziploc bag. They don't have to be a lot of soil. But one key part is that we try and clean our shovel in between soil samples so that whatever is in this soil isn't contaminating the next soil sample. And also so you're not spreading stuff around when you collect these samples. And so on Friday, I'll bring a number of buckets and with some soap water. These just have like biodegradable soap water and some brushes. And we can kind of split up and collect some samples. But generally, that's all we need. We ask, where the GPS coordinates, whether the tree is healthy or unhealthy. We're also really interested in kind of exploring some of these other species like Western hemlock and bigleaf maple, where just no one has ever looked to see if there's Phytophthora associated with these trees. So it's a pretty neat opportunity. There's also the potential that we'll find a species that's never been described or found before. So it's possible that you could collect something that a species that we haven't ever like isolated before, which would be pretty cool to have like some endemic species to the Cascades that just haven't been studied. But yeah, generally we collect soil from 4 or 5 places around a tree. You remove the litter and the duff so that you're not collecting like leaves and detritus, but you're just collecting soil and fine roots. And then in between, cleaning with some soap and water. So that's
If
a
anybody,
quick crash.
I did
Go
put
ahead.
a couple of links in the chat. So including that soil health in case anybody wants to look at it like right now or copy it for later.
Yeah, thanks, Nicole. So Nicole has posted a number of the links in the chat, which is awesome. Thank you for that summary. And yeah, we have a lot of different links. So please explore and we'd love to hear your feedback. So does anyone have any questions? I can elaborate a little bit on Sally's comment, but it would be great to hear if there's any other questions beforehand. And for those of you that I do recognize some of you, I'm glad that you're here and I appreciate that you can join us, but thanks so much for being a part of one of the previous hikes. And thanks for those of you that have actually collected and submitted samples already. It's neat to have an opportunity to share more with you. Our goal is to kind of make a story map soon so that it'll be easier and connected to the places and things like that. So you'll be able to explore that soon. and share that. So I don't know if you're going to stop recording, Nicole, and then have more of a discussion. Or do you want to record that part?
Hey,
Uh,
Joey. Oh,
Oh, I was just thinking either way, I think it depends on people's preferences too, but go ahead, Anne.
Joey, where did you say you got the seeds from in Oregon that you're planting up in Washington? And where in Washington was it you're planting them?
Yeah, that's a great question. Let's see. So the seeds we got, we actually we partnered with Weyerhaeuser. The Weyerhaeuser actually donated many of the trees. So we didn't get them from seeds, but we got them from bare root stock. And so for the trees, there was two different seed zones. One is a Willamette Valley seed zone. So the trees are from kind of the lowlands of Oregon and Willamette Valley. The other one was Lewis County seed zone. So like kind of Chehalis area. And so Weyerhaeuser was generous and donated us some trees, and it just kind of happened that we had two different seed zones. And so with that, we were able to make this experiment. So let me see, am I sharing? Sorry. There is this web page. Well, there's two web pages. Let's see, foresthealth.org/. So if you go to foresthealth.org/adaptation, I think it's in this research tab. But there you can join kind of a mailing list if you'd like to stay updated about this one specific project. But the plan was we planted them in parks because we were hoping to partner with like school groups and community colleges or classes that want to get involved. And so partnering with the class to measure the trees, then they could use the data and compare their trees to other classes. So there's actually there's actually 4 sites, but one is like inaccessible to the public. So we just have the 3 here. So one is in Discovery Park in Seattle. Hard to explain. It's kind of like, I guess this north parking lot is generally where pretty common. So from there, it's just like a little bit southwest. And so we planted them in this area. The other sites, there's one in Renton that might be a little closer. But I don't know if you need to know if you want to know exactly where they are, but there's this one in Renton. And so all of these have been really great. We're partnering with cities and parks to plant them and care for them. And so again, this is just on this web page. There's these 3 sites. Each site has 10 rows of trees, and each row kind of alternates Oregon, Washington, Oregon, Washington, Oregon, Washington. And we've measured a little bit, or we've remeasured some of them, and it does seem like overall the Oregon trees have grown up slightly faster than the Washington ones so far. But we haven't like demonstrated that statistically, so there might be some outliers that are still really contributing to that, and we need to do a little more investigation. But at least it's interesting to see that the Oregon ones are doing just as fine. And these are all just one piece. Is the other part is like who's going to survive over time. So we're still trying to track like tree dieback, tree health over time. But it's too early to really see any differences, but it's something that we're exploring because we think that it's too early to give up on red cedar, and there's opportunity to still explore some of this potential to move seeds around or try other things. Another thing that's kind of unclear is whether we're just seeing the dieback of trees that may be established during really wet periods, and so therefore never put their roots very deep. Never had to put roots very deep, and so now that soils are drying out, are we seeing all the dieback of all the trees are shallow soil or shallow roots? And if you planted a tree in that same site today, would it put its roots deeper? And in that case, would it, survive? So it's still kind of unclear whether, we're seeing whether like those sites are not conducive for red cedar at all, or whether red cedar can like acclimate a little bit and adapt to whatever site condition is present. So does that make sense?
Absolutely. Thank you so much, Joey. That's great research. I think there's a couple more questions.
Is there? Okay, good. Yeah, so Janice has asked if we prefer soil samples like while the soil is moist, or whether it's okay when they're dry. That probably has an effect on the soil, but biologically, Phytophthora, like the group that we're really most concerned with in this case, or most interested in, they can produce these spores that we call them resting spores, because they're like very thick walled. Like if the soils are drying out, it'll produce these spores, and then it will just kind of hunker down and wait until conditions are right, and then they'll actually regerminate and start growing again. So even if the soils are completely dried out, and in some cases we let the soils dry out in the lab, but when we re-wet them, it stimulates those chlamydospores, or those resting spores, regerminate and start growing again, and then they'll produce those swimming spores. So even if the soil is really dry when you collect it, we will like soak it and give it a day or two to let that fungi grow, or that Phytophthora grow, before we bait. And when we do add the leaves, we usually let them sit for 10 days or so, and that's enough time for something to start going again. But yeah, we haven't really had enough, or we haven't done a proper study where we compare like wet soils and dry soils, but that could be interesting. But yeah, your comment about like later in the summer having a better idea if it's an unhealthy tree or not, that's a good question. Again, thinking biologically with Phytophthora, they love a lot of moisture. So right now it's probably when they're like the most active and probably feeding on the roots the most. And then that could be why like late in the summer is when we start to see the dieback or the issue, because they've had the spring to really colonize and affect the roots, and then you don't really see it become an issue until the soil dries out. So it's hard to say. I think regardless, like you're always going to see less healthy trees in the end of the summer, maybe, but yeah, from the standpoint of collecting soils, I think anytime is fine, but now is a good time. Yeah. If you're just collecting from one tree, do you still need to sanitize after each scoop, or is it just if you collect from multiple trees? Thanks. So Jan has asked, really good question. Suppose you're like just going on a hike, but you don't want to carry a bucket of soap water, which I totally understand. Another thing you might do is carry like some wet wipes that have some actual alcohol or bleach, and wipe off the shovel that way. But if that's, another thing you could do is just like collect one sample every time you go hiking, and then when you're home, you wash the shovel. But I would say you don't need to clean it if you're just gonna go collect one sample, but it might be good to clean it before you collect that sample, just in case there's something on the shovel right now. If that makes sense. Like, if you use the shovel in your garden bed, and then you go and use that same shovel near your red cedar tree, you might move something from your garden bed into that red cedar, if that makes sense. Yeah, I guess with that question, I'm curious, do you feel like the cleaning the shovels is a barrier or like a huge inconvenience? Because it kind of, I mean, from the same, if you're going on a hike, I understand you don't want to carry a bucket around. So in that standpoint, maybe just collecting one is all you're good, all you need to do. Yeah. But yeah, we do recognize like,
Or just, carry like five different shovels, obviously, like you want to on a hike. No?
that could be neat if you had like 5 bags and each one had a little spade and then you just kind of cycled through. But yeah, it sounds like Jan has one tree in mind, and that's great too. I do think, one thing that would be neat is if you're going to collect like an unhealthy tree or soil underneath of a tree that's dying, it'd be neat to also collect soil underneath of a tree that's healthy nearby. And so we can compare and see if there's anything different. And so maybe on Friday, if there's time, maybe we could each try and collect a soil under unhealthy tree and a healthy tree, depending on what's there and how many of us there are and stuff. But we are trying to track, is it a healthy tree or unhealthy tree? But I think to find the differences there, we need a ton of samples, because the reality is like root is it's kind of maybe it takes time. So if you find something in that soil now, it might be 2 years from now before that tree becomes unhealthy. And so it's difficult to make those connections without like more robust digging into the roots and like sampling single trees more and more intensively, I think. Because we're just kind of like, 5 spots around a tree, trying to get as many samples as we can. And really it's like a, preliminary where if we find some stuff out there that's concerning, like then we can use that to design a more specific project or do some more tests in the lab. So yeah, this is good questions. You guys are great thinkers. CM's asked, is WASDOT included in the program?
You think it would help if people ask their questions, raise their hands, ask their questions rather than writing them in some cases?
If anyone's more comfortable doing that, they're absolutely welcome. Yeah, I think so. I'm guessing that CM has asked
that one's my question. Yeah.
Oh, thanks. Go ahead.
My name's Connie.
Hi, Connie.
So when I listen to this and I look at the trees and I think my own thoughts of watching all the trees, the Doug firs and the big leaf maples and the cedars, and trying to figure out what the situation is. It seems like it might be really complicated, but I look at the different ages of the trees and the die-offs in sort of this middle age group, or not, young, maybe a 10-year-old in a human age. And then I think, well, we've been irrigating things for a long time and now we've stopped watering. So is a matter of irrigating things as you try to raise them, which then led me to, are you irrigating your test trees to keep them alive, and are you measuring all that? Which then led me to, we've had a lot of people raising a lot of trees for a long time, And do they have information, or can you get information from them that might help? Along with WashDOT, which has planted a huge number of trees, many of which are now dying in this age group, which then led me to my own yard, where I have a cedar tree that's super happy, but it has a creek that runs right next to it. And then just across the yard, there's one that always looks sort of stressed, and it is in a drier area. And so some of those things might be also important in gathering your data. And then, okay, because you could tell already, my mind boggled, and I thought I might portray that to everyone.
Yeah, those are all really good points. You know, we've done our best. One thing that we have started looking at, and we had this really cool collaborator named Ruben Rodriguez just did this analysis with the iNaturalist data, looking at distance to streams, and so they compared the distance. Let me see if I can pull it up for you, but so that was exactly what they're kind of looking for. And it would be interesting to see, you know, how your trees do over time, whether the trees that are next to that creek end up doing worse because, you know, they may have shallower roots than the tree that's more stressed and out on its own. But this is an example of kind of the analysis that Ruben was able to do just from the iNaturalist GPS points. They were able to calculate the distances to each point from a stream. So this is kind of what it looks like, where you know, all these blue dots in this case are healthy, and then all the red dots are unhealthy. And so they were measuring, you know, where is the distance to the stream for each of these, and is there a difference? And it did kind of look like the dead trees were generally closer to the water in their analysis. So that's interesting. But yeah, you know, those are a lot of good points. Certainly we're not the only ones studying this. We hear it very often from people like WashDOT, like that they have trouble with red cedar seedlings, and there's always high mortality rates a couple of years after planting. But I'm not sure how many folks are actually tracking that in relation to climate and things like that. I would be grateful if WashDOT wanted to work with us and share some of that data. In terms of the age groups, you know, it would be neat to compare, to do some experiments or some surveys where we say we hypothesize that trees that are small are less healthy, or trees that are large. So the reason I'm showing you this iNaturalist data is there is a question on here about tree size. And so here's what we can see. Oops, I think I clicked. Bear with me 1 second. This works, it'll be kind of fun. But yeah, in terms of the red cedar planting, there is some variation in whether the trees are watered or not. Like Seattle had a water tank on site and they're planting, where Tacoma did not. But that means that we can't compare the Tacoma trees to the Seattle trees. But within each site, the Oregon and Washington trees are treated the same. So you can still compare Oregon and Washington in those sites, if that makes sense. I do agree that if we're going to water trees that we established, we need to be thinking about like not just like watering them for the first 5 years, but trying to wean them off of being watered. So maybe watering them for 3 months the first year, 2 months the second year, and 1 month the third year, and then not watering in the fourth year, if that makes sense. Because I do feel like sometimes as soon as you stop watering them, like you said, they die. So Suzanne asked if include the coordinates for any given tree. So if you add it to iNaturalist, it will include the coordinates. I'm not sure what happened to my internet. But if you're collecting soil sample, it would be helpful to know the GPS coordinate with the soil sample.
And I also want to say that the tree size is also included in the iNaturalist questions
Right.
and
Yeah,
how close it is
thanks,
to water.
Ann. Yeah, good practice would be to add the tree to iNaturalist. Like, for example, if you're going to add this one, and then collect soil underneath of it, and maybe put in the notes on iNaturalist, like soil collected on this tree. But at a minimum, we just are happy to get a soil sample where it just says like this tree is healthy and 90% of it was, dying back or something. But yeah, to go to Ann's comment, there is this question about tree size.
And I also want to make sure we get back to Sally's question.
Okay.
And
Sorry.
we've got, we're coming towards the end. It's 8:27. So just wanted to give it a little time check there.
Oh yeah. Okay, so Sally's question, I may have missed it. So Sally was commenting on the kind of the shallow, how do I say this? The shallow water table versus the deep water table. Does that make sense? So again, yeah, maybe it's possible. So there's a couple of explanations. Either the trees next to water, maybe it's more wet there, so there's more fungus there. That could be part of it. The other part is that if they're next to water, or if they're in a shallower water table, they just haven't had to put their roots as deep. And so those trees may be vulnerable to that, the water table drying out or that stream being drier, because they've just never had to put their roots very deep. And so there's kind of a couple different hypotheses there. And that's kind of what we've been trying to get at by looking at the water table depth and also looking at the distance of streams. It's really unclear.
Joey, at first you were thinking that it was happening with seasonal water. And now are you guys thinking that it's happening even if it's right next to a body of water that is there all year round?
No, well, I think that it's probably more pronounced in the areas that have seasonal streams because they probably are the most affected with longer and hotter droughts. But I'm guessing that kind of the whole system is feeling that drying out. And so whether you're close to a permanent body of water, I guess my question is, do some of these lakes also fluctuate or not, or do some of these ponds or other areas where we might see trees established, do they also fluctuate, or are we only seeing it in seasonal streams? And that might be a really good, and I think that's a good question to investigate further with Rubin's analysis on the distance of waterways. It's a good question. So I was hoping to show you, oh, there we go. Okay, so you can still see this. This is kind of the breakdown of all the trees that are shared on the iNaturalist project so far. You can see most of them are either large or most of them are medium, but there's still a lot that are small or very small. And so it does kind of seem like the trees, I wonder if that's part of my analyses. Well, we'll have to think about that more on Friday. Are we only seeing the dieback in the smaller trees or these medium trees, or are we also seeing it in small and large trees? They're good questions. Yeah, I need to look at some of my other analyses and we can explore that a little deeper.
It looks like there is over 900 of large, what you are considering large trees. So that's very interesting.
Yeah, the other, I'm wondering if there's a way to like also look at. So with this, I can't show you like how many of these are healthy and how many are unhealthy. But I could, if we have a couple more minutes, I could look at that with you. But I have to open up this other program and pull that up. So it's kind of up to you and your time. Out of respect for your time, I know it's late.
I can go either way. So if people want to stay and you want to find that information, that'd be really interesting, Joey. And then the other thing we can do is we're going to send a letter out, an email out, and we could include it in the email. So whatever works best for everybody.
Yeah, I see. Well, let's see if I can pull this up. Yeah, this might take me a minute. I think that it's too much of a struggle to pull up on the fly. I will look into this a little more. So if you can all join on Friday, or if I can give you a summary and maybe I'll try and send you a plot and that just has like tree size and tree health. But my impression is that we're seeing dieback on a lot of different tree sizes, and also it kind of depends on which part of the Cascades you're at. The west side, it seems like a lot of diebacks kind of throughout all tree sizes, but on the east side, it seemed like you're only really seeing it on the bigger, older trees, which is interesting.
That is very interesting.
It's going to take me a few minutes to really figure that out. So I think I might encourage us to break for tonight, but I do hope that we'll see some of you on Friday, and I again really appreciate everyone turning out on a Tuesday night and sharing some of your time with me.
Yeah, and if anybody still wants to sign up for Friday, I believe that the link was in the thing I sent, but I can copy it again. And we do have a few spots. And if not, you can all collect soil samples on your own. And yeah.
Yeah, even if you don't go on the hike, you can come to the hike and drop them off, or maybe in our email, we'll have a location where you can drop them. Maybe David Kapler's house, but we'll have to ask him.
This was great, though. Thank you so much for taking time to share this with us, Joey.
Yeah, I hope it was all right. Running kind of low on sleep these days, so this was I was catching myself like struggling a lot more than usual. But I'm glad I can share a little bit of update and would like to do it again sometime after we get some more samples.
We'd love that.
Yeah, we love all of your work and your presentations. And I bet aside from the lack of sleep, being a new dad is pretty darn exciting.
Yeah, we're really lucky. She's super sweet. And I appreciate the warm welcoming and the kind words. So we're in good hand. We're really grateful to have good friends like y'all, and life is just really good right now.
Awesome. Well, we'll let you go get some sleep.
Okay.
Yes.
Or die trying.
Yeah. All right. Keep well,
Thank you.
everyone. We'll see you Friday, maybe.
Thank you, everybody. Really appreciate everybody who joined.
Hi, Anne.
Oh, hi. This is
They died off so quick.